Thank you very much for the invitation and for the organizer to organize this meeting. So the title of my talk translates conflict I'm often facing when I write a paper, because usually I state that I've shown learning in slime mold, and people always say to me, no, you have just shown adaptive behavior. So I let you be judge of that, showing you my results. So the work of my team is set in the context of basal cognition. And basal cognition has been introduced by Pamela, Mike, and other colleagues. And it refers to the processes and mechanisms that enable non-neural organisms to track environmental states, act to ensure survival and reproduction. So these mechanisms include, of course, perception, learning, memory, decision-making, and other mechanisms. So if you want to know more, there's a very great special issue in Philosophical Transaction of the Royal Society in 2021. So today, I'm going to try to convince you that a cellular slime mold, also called Physarum polycephalum, is an ideal model to study basal cognition. Slime mold, a cellular slime mold in particular, belongs to a kingdom called Amoebozoa, which is a sister group of animal and fungi. You can see it here, if you see my mouse. So it's here. So it's a very, very, very old cousins of us. So slime molds in the vegetative state, called also Plasmodium, is a unicellular organism, but it's polynucleated. And all the nuclei divide in synchrony every 8 hours. So you can see here nuclei labeled with DAPI. So it's closed mitosis without cytokinesis. So you have a big, what we call also a syncytium. So this cell, when it's exploring its environment, it's moving around at speed up to few millimeters per hour. So here you can see that the cell is polarized with at the front fan-shaped, what we call fan-shaped leading fronts. So cell motility in slime molds relies on a network of tubes, also called veins. And this network distributes gases, molecules, and organelles throughout the cell. So this tube also contracts and relaxes periodically every 90 seconds, depending on the environmental condition. So if you look closely to this network, you will see the liquid phase of the cytoplasm, also called the endoplasm, that flows back and forth. This phenomena is called the shuttle streaming. The ectoplasm, which is the solid phase of the cytoplasm, contains actin and myosin, and it forms the contractile walls of the tubes, and it contracts periodically. So the contraction of the ectoplasm pushes the endoplasm toward the cell periphery, and the leading fronts extend and retract in synchrony with the shuttle streaming of the endoplasm. So in other words, slime molds are doing 2 steps forward, 1 step backward, 2 steps forward, 1 step backward, because the shuttle streaming is asymmetric. So slime molds are a very useful model when it comes to experiments, because they have an exponential growth, meaning that they double size in less than a day. And so you always have a lot of biomass to work with. And also another quality is that if you take a giant cell like that, you can cut it into pieces, and each species will give you an autonomous slime mold. So it's really, really useful for experiment, because the control in the experimental group can come from the same plasmodium. Another very useful thing is that 2 cells that are genetically identical, they can fuse, which allows you to mix, for instance, different physiological state or different past experience. And they have also a particularity which is quite interesting during their life cycle. It's when the conditions are not favorable for the slime mold, so meaning that there's no food and the substrate dries out, the slime mold would enter a dormant stage called a sclerotia. And they can remain in this dormant stage for a few years. And they are very easy to revive. So it's just like gremlins. I don't know if you've seen the movie, but you just have to drop some water on top of the slime mold, and it would grow back to life. And it would be like exploring its environment again. So slime molds live in a complex environment, which exposes them to various stimuli. And like every other cell, they possess sensory apparatus to monitor internal and external cues. And slime molds can detect and process and respond to chemicals, light, humidity, temperature, pH, mechanical stress, gravity, etc. So first of all, I'm going to give you an example of perception of chemical stimuli. So here you have a maze from Andrew Damasky, and in the middle of the maze, you put an oat flakes. The nutrient of the oat flakes would diffuse in an environment, creating a gradient. And when the slime mold will grow around the maze, it can follow up the gradients and find quite easily the food source it's looking for. On the right side, you can see another experiment showing perception of chemical stimuli is the famous cafeteria experiment, where you face the slime mold with different food sources varying in their ratio of carbohydrate to protein. And you can see that first the slime mold is exploring the environment in all directions. And once it detects the food source that maximizes its growth, it goes directly to it. So here it's twice more protein than carbohydrates. So perception allows slime mold to do decision making. And I'm going to give you 3 examples of decision making in slime mold. The first example comes from a work from Tanya Lati and Madeleine Beckman, and they show that slime mold can make irrational decisions like us and like animals. So they're sensitive to the decoy effect. So I'm going to explain. So for instance, if a slime mold is facing 2 options, one is a high quality food source, but it's enlightened and slime mold hate lights. And the other one is a medium quality food source, but it's in the dark. You will see that the slime mold doesn't prefer one option or another. It chooses either one or the other. If now you add a decoy, meaning a worse option, so it's a low quality food and it's in the dark, you will see that suddenly the slime mold express a preference for this food source, medium quality in the dark, which he was not favoring before. So this is showing that slime mold is sensitive for decoy effect. Another example of decision-making experiment is, for instance, something that we shown in 2019, that slime mold are sensitive to the speed versus accuracy trade-off. So meaning that fast slime molds are less accurate than slow ones. So here you have 3 different genotypes, GPN, USA, and OS. And here you have the accuracy in a task. The slime mold needs to find the best option in a little cafeteria. And here you have the decision time. So in fact, what we show is that the slime molds that are very, very fast at answering these tasks are usually getting wrong, or they act randomly, while the slime molds that are making very slow decisions are very accurate. The last decision-making experiment we were interested in is the Weber law. So we show that slime molds do not follow a Weber law, actually. So they're doing everything like animals except that. So for people who doesn't know the Weber law states that differences between 2 options are perceived in relative terms and not in absolute terms in all animals. So for instance, here you have an example with 2 M&Ms pile, and the absolute difference in the first example on the top and the second example on the bottom, it's exactly the same. The absolute difference is 8 M&Ms. But if you look at the 2 tasks, the second one is far more difficult to solve than the first one, because we are sensitive to relative difference and not absolute differences. So as the Weber law predicts that if the absolute difference is constant, the performance will decrease with the stimulus intensity, so the number of M&Ms. So we tested that with slime molds. So we ran 82 different binary choices that we repeated many, many times to cover a lot of space. So here it's a stimulus intensity. So here in our experiment, slime mold had to pick between 2 different concentrations of sodium chloride, something that they don't like. And so we increase the concentration of sodium chloride. And here it's the absolute difference between the 2 options. And if you, for instance, pick one absolute differences equal to 2, and you follow the line, you will see that the performance doesn't decrease with stimulus intensity, but it actually increases. It goes from 0.7, between 0.7 and 0.8 to 0.9. So in slime mold, differences are perceived in absolute terms and not in relative terms, which is very, very different from animals. The second experiment I wanted to highlight in slime mold after decision making is habituation, which is a non-associative form of learning. So habituation is defined as a response decrement to a repetitive stimulus, and it is an ubiquitous way to tune out irrelevant stimuli in the environment. So the task is very simple. The slime mold has to cross a little bridge to get to the food source. And we measure the first pseudopod, the latency to the first pseudopod, and the time to cross the little bridge. And in this little bridge, we put some more chemical repellent. So we tested different repellent, quinine, caffeine, vanillin, called sodium chloride. And once it crosses the bridge, we transfer and we ask the slime mold to do it again. So here's the result. So First, habituation is a response decrement to repetitive stimuli. So here is the video at the bottom where you can see that fly mode has to cross a bridge with quinine. So you have a control group, and after you have the multiple stimulation. You will see straight away that the fly mode go quicker and quicker when the stimulation is repeated in time. And so here on the graph, you can see the day. Here you have the habituation index. The habituation index, when it's 0, it means that the fly mode is not habituated. When it increases, it means that the fly mode get habituated. So we have a nice habituation to quinine. But habituation responds to different criteria. You cannot just show that to show habituation. You have to check a lot of criteria. The first one is specificity. Specificity means that the response decrements you observe show a stimulus specificity. So for instance, after 6 days of being habituated to quinine, if I face the fly mode with caffeine this time, it will show a strong reaction toward caffeine. It is specifically habituated to quinine. The third criteria is recovery. If you remove the stimulation for 2 days and you test the fly mode again, the response recovers. The fly mode reacts again to quinine. The fourth criteria of habituation is stimulus intensity. So the more intense the stimulus, the less rapid the behavioral response decrement. So it's true that if you increase the quinine concentration like that, you will need more stimulation for the fly mode to get habituated. At 10 millimolar concentration of quinine, we don't have data because the fly mode didn't want to cross the bridge at all. The fifth criteria is dishabituation. This criteria is actually quite interesting because this was never demonstrated in other unicellular organisms. So dishabituation is the presentation of a different stimulus that results in an increase of the decremented response to the original stimulus. So for instance, you have here the day, here you have the habituation index. If I habituate a fly mode to sodium chloride and I leave it alone for 1 day, I test it again, it's still habituated to sodium chloride. If I do the same experiment, but this time, instead of leaving the fly mode alone, I face it with vanillin, another repellent. We can see that the answer to sodium chloride decreases. So this is a dishabituation. The last criteria we tested very recently is potentiation of habituation. So after multiple series of stimulus repetition and spontaneous recovery, the response decrement becomes successively more rapid and more pronounced. Indeed, when you do it again and again and again, you can see that on week 1, the habituation is okay, but on week 4, it's far better, it's more intense, and it's quicker. So we have this potentiation effect. So due to the fact that fly mode can go into a dormant stage, we can test things that we cannot test with animals. So for instance, if I habituate a fly mode and I put it in a dormant stage, what happens when I revive it? So this is something we did. We take habituated fly mode, we put them in a dormant stage. We did the same with control fly mode who were not habituated. And when we revive them, we tested for the habituation. And indeed, the habituated fly mode went to a dormant stage for 1 month or for 1 year, were still habituated to the chemical repellent. Then, because we cannot do that even with animal, we tested how fusion works when it comes to learning. So if you have a fly mode that is habituated to caffeine and a fly mode that doesn't like caffeine, you make them fuse and you test them again. Do they both like caffeine? So we did this experiment by doing chain of transmission with, for instance, 1 naive fly mode, 2 naive fly mode, 3 naive fly modes with an habituated fly mode. And we tested the fly mode, and what you can see is that if you make them fuse for at least 3 hours, the habituated fly mode, of course, remain habituated, but now the unhabituated fly mode is also habituated. That you can see on this video, they have exactly the same behavior. You cannot differentiate an habituated one from an unhabituated one. So this gave us an idea of the mechanism. Something must circulate inside the cytoplasm that the fly mode can transfer to the other one. And the most parsimonious hypothesis was that the fly mode was absorbing the repellent and sharing it with the other fly modes. So what we did was to measure the amount of sodium chloride inside the fly mode for habituated one and control fly modes. And what we show is that habituated fly modes contain 30 times more sodium chloride than controls. And in the dormant stage, we found that habituated fly mode contained 10 times more sodium chloride than unhabituated fly modes. So then we had this idea. So what about if I inject the sodium chloride directly inside the fly mode? So I take a naive one and I do a topic injection of salt inside the fly mode. Would it be habituated then if it's only about the chemical repellent? So we did that. We had 2 naive fly mode. One was injected with sodium chloride, one with water. water. And you can see that the ones that was injected with sodium chloride show a higher habituation index. However, the habituation index was not as good as a slime mold really trained day by day with sodium chloride. So we keep digging into the mechanisms. And recently we looked at the topology of the network, and we show that the slime mold that has been habituated to salt is building thicker veins than a slime mold is not habituated. So the vein was significantly thicker in habituated slime molds. Then we looked at gene expression, and we show that acting mRNA expression was 30 times more in the habituated slime mold than the unhabituated slime molds, meaning that the slime mold was really investing in its network of vein to push stronger to counteract the repellent. Now we have been designing software to look at the oscillation, because you have oscillation of the cytoplasm inside the cell, and we are very interested in this pattern of oscillation and to see if they're correlated with learning. So this is an ongoing thing. So studying learning, of course, we were interested if learning performances decrease with age, like you can see in animals. And so what we discover is that not at all. A slime mold that is very, very old can still learn very, very well to habituate to a repellent. However, what we noticed is that the speed itself of the slime mold is really, really significantly affected by the age of the slime mold. A slime mold that is 2 years old is far slower than a slime mold that is 1 month old, for instance. And this difference in speed is amplified when the substrate contains a repellent. So building on the learning experiment, we were wondering what happened after dormancy. So what we did, we took a young slime mold, 1 month old, a 2-year-old slime mold, and we put them in a dormant stage, and we revived both of them. And we noticed that the old slime molds, at least for the weeks that we tested at the dormancy period, recovered its speed, which was quite interesting. So we were like, what's happening during dormancy? So the first thing we wanted to look at is oxidative stress, because we know that it's highly correlated with age in animals. And so indeed, we found that using Metersox threads, that the oxidative stress is increasing with age, but after dormancy, it's decreasing. So the cell is kind of doing a cleaning up during the dormancy period. So what about cell fusion? Is it like learning? Can you share your age with the other slime molds? So we took a young slime mold, 1 month old, and a 2-year-old slime mold. We let them fuse, we separated them, and we tested them independently. And what we show is that the young slime mold at a speed that was slightly affected by the fusion, but the old one really kind of recovered by fusing with a young one, which was quite interesting because it says that you have a benefit to fuse with a young one. So we knew from previous works that we conducted in our labs that slime mold, they can release chemical cues in the field, and these chemical cues are used by the other slime mold as cues. So for instance, we have shown that slime mold move toward chemical cues left by well-fed slime molds. These cues are in fact calcium that slime mold is releasing in the environment. But we also show that slime mold move away from chemical cues left by starved slime mold, irradiated slime molds, or poisoned slime mold. So we were wondering what happened with aged slime molds. So we did this very weird experiment where we put a young slime mold, an old one, we left them for a while in the environment, we removed them, and we put a third slime mold. This first slime mold could be young or old. We tested both of them, but in all cases, they preferred to go towards young slime mold chemical cues. So slime mold could be able to differentiate age from a distance, which was quite interesting. So based on this result, I hope I've convinced you that slime mold are an excellent model in which to identify the mechanism mediating cellular decision-making and learning, or if you want to call it, adaptive behavior. But my presentation doesn't stop there. As David said, we have to think outside of the box. And recently, I have been trying, because it's not everything is not about doing research. We also have to share research if we want people to listen to us. So recently, I've been involved for the last 2 years in big citizen science project with slime molds that I wanted to talk a bit. So the first one was called Élève ton blob, which is a play with word in French, not in English. And the question was, does microgravity affect slime molds' behavior? And this was a project with the National Center of Spatial Research and the CNRS and the Academy of Toulouse. So the goal of this project was to increase the young public understanding of science and research, promote scientific careers, and move scientific content from the abstract to the tangible. So we had 5,000 schools, meaning 350,000 school kids and a French astronaut in our experiment. And the experiment, we had two experiments. Foraging experiment was a slime mold needed to build a little network with 4 oat flakes. And another one was a slime mold just needed to explore its environment. And so everything was in the box because the European Space Agency was very scared of the slime mold escaping from the box. So here are the results of the thousands of schools and the International Space Station. So the first thing we show is that the latency to move to revive was shorter on board the International Space Station, and the slime mold were quicker on board the International Space Station. And something which was actually quite interesting, if you look at the video, it's very bad quality. I'm very sorry, and I think it's worse over there, but if you cannot see it, you have to believe in me. At one point, the slime mold would kind of just stand up. As the kids say, he's saying hello from outer space. And actually, the slime mold is not limited by gravity anymore. And so it built these 3D pseudopods that are floating around, which was kind of cool. But the experiment was supposed to last 1 week. And in fact, it lasted a year because kids got crazy about slime mold and they started to do all these experiments. They even asked me to do a dictation. So I did a Zoom dictation with 300 schools in France. And it was like how to hunt slime molds. And we did also a kind of scientific experiment, a crazy one, where we met in a center of France. And I asked the kids to bring back the slime mold that I gave them at the beginning of the year. They were all clones. And so what we did was to put all the clones next to each other and build the longest cell in the world, which is 53 meters long. We even invited an expert to take a picture of all the fusion events to be sure that it was a single cell. So I don't know if in a learning transfer experiment, it would work with such a giant cell, but it was actually quite an interesting phenomenon. And so the last project I run, it was just last year, it's still an ongoing project. We wanted to observe the effect of climate change on slime molds. So the goal of this project is to raise awareness about climate change, increase the public understanding of science and research, and develop critical thinking and explain the scientific methods. So I don't have to talk about the global warming. I know you're all aware of that. So in the last 80 years, there was a plus 1.2 degrees. And the prediction for the next 80 years, optimistic one plus 2 degrees, pessimistic one plus 8 degrees. Plus 2 degrees, 5% of species will go extinct. 4 degrees, 16% of species will go extinct. But somebody says, nobody's talking about microorganisms. Everybody are focused on animals. What about microorganisms? They're fundamental in the ecosystem. So we decided to launch this experiment. So we had 48,000 applications and we selected 15,000. When I say we selected, in fact, the slime mold selected them because I attribute randomly numbers to the participants and the slime mold picked the group he wanted to work with. And so we had nursing homes, schools, families, students, etc. So we had to prepare 60,000 samples for all these people. It took us 8 months with Céline and Emily. It was a huge job. And the people had to buy a specific list of material. And we, of course, set a material exchange tools was called GoGo-Carto. So the people could exchange material to limit the ecological impact of our experiment. So the people were looking at the duration, the intensity, and the frequency of the heat wave. We had 15 different profiles, and each volunteer had a control and an experimental group at home. And they repeated the experiment on 8 individuals. So to train the volunteers is like having 15,000 interns, but they're not in your lab. So you have to train from a distance. So we did a written protocol for mainly old people and videos for the young people who don't want to read. We had quiz to check if the people understood the protocol. We had like a Facebook group, videos, webinar, and I exchanged 20,000 emails with the volunteer only in 2022. And so the data we collected from the volunteers was exploration behavior and the growth of the slime mold. And we collected all the temperature, and we have, for now, 800 and almost 1 million of pictures. And the people were also asked to do the data analysis to draw the contour of the slime mold in the Petri dish. This to feed a machine learning algorithm that would detect all the other slime mold in all the Petri dish of the experiment. So they were very interested in this machine learning. So I don't have results to show you for this project because it's ongoing. The first result will come out in July. So I want to thank all the people who work with me in the lab. So colleagues, of course, students and postdocs who are doing most of the experiments, which is the habituation experiment are quite tenuous to do. So really, I want to thank them. And I want to thank the 2 teams who work with me in the citizen science project, and especially, of course, the people who participated. Thank you for being a bit late. Sorry.
Thank you very much for your talk, a very entertaining talk, Audrey, and we have time for a couple of questions. Eric, could you go to the microphone?
So that was really, really, really interesting. I'm just curious if you've looked at whether or not the other repellents actually get absorbed by the cells. So sodium chloride, I can imagine, look, it will diffuse through the membrane, so it's easy for the slime mold to pick that up. But I don't know about caffeine. I probably should, because I should know some biochemistry, but I don't remember if it goes through a membrane. But did they, or like quinine or whatever, do they pick that up too, or is it more a different mechanism in those cases?
We didn't measure. But the only thing is at the behavioral level. At the beginning, we wanted to see if slime mold, could share chemicals. So one habituated to sodium chloride, make it fuse with one habituated to quinine, and see if they knew and if they could be habituated to both. And in fact, the slime mold that was habituated to sodium chloride didn't want to fuse with the one habituated to quinine. So they kind of come in contact, but the fusion couldn't work. So I think that quinine is at least on the membrane or somewhere because the slime mold was itself a repellent. But we didn't check for caffeine and quinine. To be honest, we did sodium chloride because it's an easy chemical to measure inside a cell, but I didn't know any kit to measure quinine and caffeine.
Okay, other questions? Is there a remote question? Okay. All right. I have a question, Audrey. Do you have sensitization? So you have dishabituation. Do you have sensitization in the slime mold?
Yes, we show evidence of sensitization, but we didn't check all the criteria left. But yes, to solve certain concentration, we have for instance, if you use sodium nitrate, for instance, you have sensitization. Yeah. But I have to check all the criteria to be
yeah.
Poor.
Okay. All right. Well, thank you very much. Merci beaucoup and bonne journée. If it's maybe it's evening in France.
But. Yeah, it's already 8:30.
Oh yeah. Okay. All right. Okay. Bonne nuit alors. Thank you, Audrey.